I'm delighted you're here, and I'm happy to talk to you about clinical trials for rare diseases. And so, in my field, as a trialist, I'm part of a group that's a bridge between the research community and patient care. How do things get out of the lab into the bedside? How does that transition occur? And the accepted methodology is to conduct a clinical trial. And so it's not easy to do clinical trials. And the principles underlying clinical trials are the same whether we're talking about a rare disease or a very common disease. Really, they're essential trials, essential constructs that are absolute. And what needs to be said is that even more so, there's a mindset in terms of being a successful trialist, being able to successfully to imagine, create, and implement a trial. And that's one that's very difficult for us all to maintain because, the drugs we get to work with and the treatment approaches are really the product of our passion and our hope that what we have will improve outcomes. But when we're actually engaged in a trial, we have to be in equipoise. So we have to say to ourselves, we really don't know which treatment is best. But we didn't get there thinking that way. We got there thinking that we might have something that helps. But otherwise, if we really believe that our new agent, our investigative agent or approach really helps, then it would be unethical to randomize people to anything but, give them anything but the treatment that we believe in. So to actually enroll participants, we have to understand that we don't really know. That's where we are when we design and conduct a clinical trial. We really don't know what's best. And the other part of that is I think we need to understand the sacrifices that are made in the conduct of a trial. And these are sacrifices made on the part of the investigator and the participants in the trial. Both are making considerable sacrifices to enable us to answer important questions about outcomes and how we might improve that. And we should never forget that our participants in the trial are very much our partners. And we really ought to talk to them before we launch a trial and they understand the burden and we understand the commitments that both parties have to make to be successful in such a trial, in such an enterprise, to be successful. And we have to realize that, from the participant standpoint, they're participating in the hope, that, they will answer a question, if not for themselves, for other people. The altruism is very important to participate in a trial. And in part of the investigators, we're passionate about improving those outcomes. But we have to accept patience. We have to realize that it'll take time and we won't get an answer. So it's very different than treating a patient in clinic where you have, immediate, if not instant, immediate gratification, to conducting a trial where you're going to treat and you really don't know whether you're helping or not. And sometimes you won't know for years. So we all have to make sacrifices to engage in this enterprise. And it's not so easy to do. So I think that when we talk about clinical trials for rare diseases, we need to focus on, what are the constraints that we have to live within? And clearly, rare means small numbers. So we really have to figure out, are there better ways to study interventions, alternative strategies, when you only have a small number of patients? Now, I do this, but I want to point out that while I have been in the field, rare diseases, for quite a long time, and, but what I usually hear is that we need to band together because cumulatively there are 6,000 or more rare diseases, and cumulatively our voices are much louder. But I really think that almost every disease will be a rare disease. Science is moving in such a way that, we won't be talking about cardiovascular diseases, we won't be talking about leukemia, we won't be talking about breast cancer, but in fact, we're going to dice it into smaller and smaller slices as we learn more and more about these diseases. And those smaller and smaller slices are going to look more and more like rare diseases. So I think we're in a transition to where what we've learned how to do studies with small numbers of participants will be in the lead. People will come to us and how to do these kinds of studies. Clinical trials are expensive. Clinical trials that enroll lots of people are very expensive. If we can answer a question with a smaller number of people, then everybody will be a path to our door. Everybody will want to know how to do that because, that means you can do more studies, you can do them more quickly. Everybody will learn from us. I think the future is in our direction, and we need to hold on to that and continue to demonstrate that there are ways that we can do trials with smaller numbers of people and be efficient in doing that and teach others who, think they can enroll everyone with, in one of my fields, in type 1 diabetes, or in everyone with type 2 diabetes, or everyone with osteoporosis, when in fact we'll find out that those are big labels that really cover a very heterogeneous group of diseases. And in fact, by cutting them into smaller slices, we'll have better targets, better intervention strategies, and better outcomes. And I think that this rare disease community is a role model for what's coming. And so I'm really encouraged and like working in this space because I believe that is the future of medicine. So to get back to the real issues here that we face every day, we wanted to look at new therapies. We want to repurpose existing drugs. We want to test new biologics. We want to make advances. And the paradigm that we live within is a clinical trial. So we have to figure out how do we adapt the clinical trial that meets all these standards and criteria in such a way as that we can be successful and answer a question. So I thought that, we need to take into account sort of what we're dealing with in the context and what are the constraints. And certainly, we need to design trials with small n. We need to understand that it's hard to enroll people when the randomization involves a placebo arm with people who are affected with the disease. That has significant consequences in terms of their everyday lives. So we have to think, are there ways around that we can really make significant progress? And so I thought that we should really talk about different designs. We've had an introduction to some of them today. Leanne talked about platform designs. I'm going to tell you how we can adapt that to rare diseases, very small numbers, to meet some of our constraints, but still be highly efficient in this process doing that. And then talk about, what are our options going forward so that we can make advances and really, show the larger scientific community that, they need to come to us. They need to be here at this meeting and understand how you can do trials with smaller numbers, right? They need to seek us out as opposed to us continuously knocking on their door saying, wait, you need to listen to us. We're an important constituency as well. So let's start at the beginning. And basically the traditional model, actually several of the trials that were presented here today are what's called parallel group design. You enroll a person and you assign them, randomize them to some experimental treatment or to some standard treatment. That standard treatment is the control, and in the cases where there is no standard, that might just be a placebo comparison. Now there's pros and cons of using a placebo in a trial. One is that, the placebo will have, what we think, no effect on the outcome of the study. And any intervention then that does have an effect has a big effect size. So the bigger the effect size, the smaller the trial. If we run the trial where the control arm is a standard of care, where there's actually treatment, then that delta could be much smaller. That makes for a bigger trial. So we have to be, very insightful in terms of how we design the trial and what we pick for the different arms of the trial as well. But typically we would enroll, we would treat, we would assess an outcome, right? And that at the end of the day, when the last patient is enrolled and the outcome for that patient is observed, then we can compare the two arms and decide whether we've made a difference. Now, most of the statistics that are presented that actually you saw on slides earlier today compare the average outcome of each arm, and they compare the average response to the therapy or the control and make a decision whether there's a significant benefit from the experimental intervention. So I want you to keep in mind this term average, because you'll see that, that's pretty limiting. If you stop and think about it for a moment, the average, there'll be some people that respond very well, and there'll be other people that aren't responsive. Can we take advantage of that? Can we learn something more than just comparing averages? And maybe we can, and I'll show you how to do that in a minute as well. So we can get more information out of a trial than what you typically see reported as an outcome, because that's what they're reporting. They're reporting the average number of new lesions, the average, patient-reported outcome, the average response. But I suspect that we really know more about that, and when we see patients in clinic, we see this all the time. There's a gradient, there's a variability in response, and we need to think about, can we learn from that in a way that helps us? So I'm going to talk about that as well. But you can see that this is very sequential, right? So I have a period of time that I enroll people, treat people, and then I wait for the outcome. If it takes several years to enroll people, then I have to wait several years for observe that outcome for the last person enrolled. That makes for very long trials. There might be a better way to approach that and think about it. And so in terms of designs, we should, look at all different kinds of designs that are possible. We shouldn't just restrict our thinking to parallel group designs. We should really be thinking about, what other things that we can do that might enhance our ability to learn from our patients in a study situation. And so this is just a long list of things that we have in our back pocket as tools when we understand the questions to be asked. And so there's a common thread here. See if I can do that. Yep. So the typical parallel group design is, one patient, one treatment, one outcome, right? So we don't have to do it that way. Maybe we have one patient, so that's the limit, but we can have multiple treatments and have multiple outcomes. So we're going to use some ideas and tricks of the trade here to multiply our experiment so that we can use information from one person multiple times in a trial. So we're going to take multiple measurements from that person. We're going to treat that person multiple times. We're going to multiply our population in such a way as that we can learn more from a single person, because we have a very limited number of people that we can enroll and play with. So that's kind of the thing to look at. And so when people come to me about designing a trial, I think about, what trials work best? What kinds of designs work best for what kinds of setting do we have? For studies with very limited patient numbers, I take one approach. When there are multiple treatment arms to consider, I take another. So Leanne described, we have multiple treatment alternatives, and in some disease settings, we just don't know which of them is any good. And in clinic, You know, you start with one, and if it doesn't work, you move to the next. If it doesn't work, you move to the third one. So yes, it's a very common situation where we have a lot of alternatives, and you know, we don't know how to evaluate which ones to start with there. And you know, there's the point that I just raised before. You know, placebo-controlled trials are difficult because you know, everybody who participates in a trial wants to benefit from that trial. You know, and so there's a lot of reluctance, as well as our usual model when we go to our physician for help. You know, we expect some treatment, you know, as opposed to being told, well, I'm going to flip a coin and you might get treated, or I might give you something that really doesn't do anything. I think that takes a lot of trust in a building relationship to try to figure out how to do that. And are there ways to overcome that barrier to enrollment? Because that's what we're really looking at here. Now, to start, if I talk about limited patient numbers, there are a couple of standard approaches that we use all the time. One is a crossover design. So you enroll a person, you treat them with A versus B. So we try to make it all double-blinded, so no bias here. A could be the experimental intervention, B could be the control, or vice versa. Usually the patient and the provider are blinded to that, so they really don't know. And then after a period of time, you know, the person who received B then gets A, and the person who received A then gets B. That sounds pretty promising, and that gets me 2 observations on a single patient. It makes the study last a little longer. I have to worry a little bit about that. But the problem with that is that it's not very successful, because if you're a patient or your patient received A and is doing well, they won't want to switch to B. And that undermines this whole design. So only the people that are doing poorly will want to make the switch. So that introduces this kind of bias. So these designs are really problematic. In my experience, really don't work very well. Now there's a factorial design, which is a different variant of this, which is actually more fun to play with, because you know, we can look at 2 different treatments and essentially we can double randomize. So if we have treatments, you know, A versus B, let's say, you know, A versus control, and we have another treatment of B versus control, we can randomize that individual on entry twice. So what they get is A and control, B and control, A and B, right? Those are the options that are there. So analytically, if there's no interaction, and I need to find that term, it came up in discussion, I was interested to hear that. If there's no interaction, we can evaluate A versus control and B versus control as if we were running 2 different studies in 2 different populations. Because if A is better than control with B in the background, and it's better than control with not receiving B in the background, then I can combine these arms analytically, and I can still see at the end of the day whether this experimental intervention is better than control. So I can get 2 for 1 out of this by doing that. Now the problem is, you know, what if A is better than control when the patient has not received B, but A is worse than control if the patient received B? That's the definition of an interaction. So if it goes in the reverse direction, then we have a problem. So we have to analytically try to adjust for that. And we have to assume that, or we can test for that and see how big an effect it is. And sometimes that'll drive up the sample size when we have to take that into account. So there's a price to pay for that. But in fact, if you know, we have 2 different mechanisms, as was described earlier, you know, one's an antibiotic and another is ventilation, 2 different approaches, one might argue that that interaction, that risk of interaction is very small. And the benefit in one group might be a little smaller, but if it's in the same direction, we can still take use of that. And essentially, we get double the bang for the buck. We get twice as much information out of that same patient by doing just a double randomization. And then there's also something called a sequential multiple assignment randomized trial, a SMART trial. I actually came up with the acronym before we came up with the name for the trial. So, and here the idea is that, if you have a trial and you randomize someone to an intervention, an experimental arm versus a control, and then you can measure the response in the controls, then they're available to re-randomize them because they didn't receive any other agent. And so, say a one-to-one randomization, so half of them will get the experimental intervention. So you've gotten more mileage out of that group, right? So there's an opportunity to take advantage of the relationship you build with the participant, and actually it does extend the trial, but you actually conduct two trials in one person because the control, as I mentioned already, we assume doesn't have any impact, doesn't have an effect. So we can re-randomize those people and take a look at their outcome. So that's an enhanced design for that. And then, I'm going to offer a new one for you, if I can make this. There we go. And so there's a new development. Actually, it's fairly new since I was here last year presenting to this group. I should digress for a moment here. So, as I said, I'm really flattered to be back and asking to talk. So I gave a presentation last year, and I had a lot of trouble getting here. The flights were very delayed, and I ended up flying all night, getting here the morning of the meeting. And in my foggy memory of that day, because of the jet lag, I wasn't really sure whether my presentation was well received, or coming back this time, I'm just given a second chance to do a better job. So I don't know, you'll have to tell me or tell the organizers at the end of the meeting. But in any case, the point being here that, what we could do is design a trial and, analyze the trial in the kind of interim partway through, and then decide if the results are promising enough to continue. Now, the traditional way we do this, is what I'm going to show you in the left and the right. So let's start with the left. So the left is what we would call analytically, statistically, an early emerging difference. So in trials where it's possible to do this analysis, look at outcomes during the trial, we could evaluate and see whether there's a benefit to the experimental agent versus the control. And if that benefit is large enough, we might stop the trial. We declare victory and go home. And so I provided the references for this. And essentially what this means is, if I plan the trial for type 1 error, statistical significance of a p-value of 0.05, 5% type 1 error, and partly through the trial, I see that the difference is 0.001, then I might say, hey, let's stop. And essentially, those lines that are converging there, I wonder if I can do that. Yeah, so these lines are the boundaries, and they're converging to a point which is, that interval is that 5%. That's where the error is. And this line reflects how much information we have. So if we're halfway through a planned trial, which puts us about here, we can calculate the distance, either positive or negative, one direction or the other, it's directional, that we observe. And if we see evidence that it's greater than these criteria, then we might say, we have enough evidence to declare victory. We have enough evidence to show a positive effect, and we can stop the trial. So there is a way of monitoring for what's called early emerging differences, and interim analysis should be built into trials that take a long time because of a chance to look at the data. There's a bit of a cost here, but I won't get into those statistical details in doing that. But nonetheless, this is very common and a standard practice to look at. Now, the other side of the coin is true also. Suppose we get halfway through a trial, so how far we are from left to right is how much information we have based on the expected population we're going to enroll. And we can look at the data and we can say, well, suppose we look at the power of the study, and then we assume that the alternative hypothesis, the benefit, the effect size that we hypothesized is true, and we have this many more patients to enroll. What is the power of our study? That's the type 2 error, right? So we typically design a study with a type 1 error, a p-value, and a type 2 error, power, statistical power. And so we can calculate what's called the conditional power. We can look at what we've got to date, and then we can just assume that what we test or designed our study to detect, the power, is true. How much power do we have? What is the probability if the difference we hypothesize is actually true? What's the probability we would see it if we continue the trial? So that's what this dashed line is, and it goes up because I'm assuming that the alternative hypothesis is true. And somewhere it'll cross this axis, which essentially is the power of the study. So the point being here is I can look at the data, and if I have two survival curves, let's say, or two estimates of an outcome, and they're right on top of each other, then I might argue that if I enroll the other, say, half of my patient population, those two things are not going to come apart. I've already got so much evidence that they're going to be the same. It's time to stop. And that's called a futility analysis. That's what that is, right? So this is black or white. We decide when we're going to do this based on the information we have, and we decide, is there enough information to stop, or is there enough information to continue? So there's a variant of that I wanted to chat about, and it's called promising zone design. And so what it does is it says, I designed the trial with certain parameters, certain assumptions of what the effect size that I'm looking for. So I might sit around with my colleagues and we'd have a discussion, including patients, and to say, well, how big an improvement is meaningful? And we pick that improvement and design our study to detect with adequate power. But we do the trial and we see, well, we're not getting that size effect. But we are seeing something. So it might be worth extending the trial, increasing the sample size, and see whether we can see statistical significance, whether we can conclude a benefit. If I don't extend the sample size, then I'll be risking the chance that maybe I expected a 50% improvement, but I actually measured a 35%, but then I'll have to conclude it's a negative study. But if I added more patients, then I will have power that if the true difference is 35%, it'll be statistically significant. So the trade-off we have here, especially for diseases in which we have no treatment at all, might be it's worth continuing because maybe our treatment is beneficial, not as much as we would like, but will be beneficial, and then the next study can build on it. So it's rather different than saying, let's take an all or nothing approach. We're invested in the trial, we're seeing a result, and it may not be enough to stop the trial, either for futility, for early benefit, but it might be enough to say, we really ought to do a definitive experiment here, so we should reestimate the sample size, and try to see whether we can find a treatment that is better than control. Maybe it's not as much as we would like, but if it's significant, it gives us something to build on. And that's very appealing when we have nothing as a standard therapy. We have nothing to use here. So this is fairly new, and conceptually, all these designs take a lot of thought and a lot of discussion to make sure we understand it, we accept it, and we're willing to commit ourselves, which is why I started out with the idea that, it's a discussion before you start in terms of doing a study. But that's something that we ought to consider, and this is an approach that is worth doing, and it's especially important because we can design the trial for the larger effect size. So that usually means a smaller number of people. And then if we see something promising, we'll increase the sample size. So you have a lot of information. One is what your accrual rate is, so you can see how feasible it is. How much are you extending the study to get this potential benefit? Whereas if you started out with a smaller effect size, it becomes a much larger study. Might be hard to get funding for it. Might be hard to, get a accrual rate that's satisfactory. A lot of unknowns at the beginning of a study. But now that you're partly through it, you have really good idea of what your accrual rate can be. And it might be practical then to go ahead and look for a smaller effect size. Whereas, as I said at the very beginning, before you launch the study, you're not really sure what the acceptance rate is, what the accrual rate really will be, and whether the study is feasible at all. But you have a lot more information at this point. It's something to think about when you think about designing studies going forward. Now, what about the next case when you have multiple treatment options and, what are you going to do with that? And so, the issue there is that, to set up multiple comparisons is really going to drive the sample size to make a very large study. That's very hard, and we come up with clever ways to avoid that. So if you had, a control, and then you had two agents, let's say, that A and B, then the question might be, well, one way to avoid the penalty for multiple treatment arms is that you decide to do just two pairwise comparisons. So control versus A, control versus B, but you don't compare A to B, because then you get involved in multiple comparisons and there's a penalty for that. So not only have you added another treatment arm, but overall there's about a 25% penalty that you have to increase the sample size because you have to make one more comparison, And then likely, as I mentioned earlier, the difference between treatment and the control is likely to be a bigger effect than the difference between two active agents or two experimental agents. So if you really want to see the same effect size, there's a penalty in terms of accrual. So we do this, but it's problematic. Enhanced designs, I mentioned a little bit about smart designs. They're enhanced designs or approach that are worth looking at as well. But now I'm going to deliver on my promise. I'm going to talk about platform designs as another strategy. Now I'm going to talk about them in the context of rare disease, because I think that's really fascinating and some things that we can do. So let's start with, a slightly different description of a platform design than we heard about, think about. So the first one is, we've got 3 time periods, and we're going to enroll in each time period, And we're going to follow patients who are enrolled in each time period to the end so that we have an outcome for that drug. So very much different than what Leanne talked about. My concept here is, here I have a fixed sample size. Not I have a unknown sample size and I'm using the adaptive randomization to continue to accumulate data until I have a winner, because those kinds of designs theoretically will never end. This design guarantees an end. And I'll show you how we play this game. And by the way, I'll try to make the point, so if I don't, remind me to do it. But it also provides a very important link between registries and trials. Because you have to think about, what is the value of a registry? What can we get out of it? And how does that help us? All right, so let's figure out what this is. So I've got 3 trials here. You know, it's really simple. I'm going to do a trial in each time period, and I'm going to accrue, in this case, 92 people exposed to drug 1, 92 people 1-to-1 randomization control. Each time period, I'm going to do the same thing. Well, we do that. You've heard about studies designed exactly like that that were presented, and then you've heard about them. Consider that period 1, and then they want to do the next trial, so they're going to move into period 2, and they want to do the 3rd trial. Well, that's pretty inefficient. Actually, we can do better than that. So let's figure out how we can optimize that. So one approach is that as you can see, we have a bunch of different placebos, patients enrolled and treated with placebo, right? So now we can think about, why do we have to continue to enroll to the placebo arm? Why can't we share the placebo arm against multiple intervention agents, right? Why can't we take advantage of that group and not have to re-enroll more patients and assign them to placebo? Now, we have to make some assumptions here. We've got to look at that, and you can see from my green arrows that, if I want to use the patients that are enrolled in period 1 to the placebo arm, when I compare it to, in period 2, to the second drug, somehow I have to make sure that, that they're comparable, that they're a valid comparator to the second drug as they were to the first. If they are, then why not? So one form of a design is really to continue to share the placebo patients against multiple experimental agents. So I don't have to enroll past the first 92 placebo patients. And, therein lies an approach, and it sounds really good, but we do have a little bit of a problem here, and that is, can we use all 92 of them from period 1 into period 2? They were enrolled at a different point in time. Maybe that, individuals who accepted the trial drug 1 versus placebo are different than the patients that would accept the trial of drug 2 versus placebo. Drugs are different. Populations might be different. So what we do is, So what we do is we share the placebo patients. So we do this borrowing. And so we take half of the placebo patients from period 1, drug 1 comparison, and then we enroll the other half contemporaneously with when we enroll in drug 2. So that, we kind of balance this bias that might be there because drugs 1 and drugs 2 might be very different, and their acceptability might be different. And then as you can see here, I extended it to 3 as well. And so what I've done is reduce the number of patients I need to enroll to do successive trials. So my point being here is, we have to think to the future. We shouldn't think about each trial as standalone and we're done and then we start over again. what we ought to do is think about the fact that there will be another trial. How do we use that information in our design to answer our study questions in a way that's really more efficient? So this one I'll give you is one that I'm doing right now. So this is in type 1 diabetes. So type 1 diabetes is not a rare disease. However, finding people who are at risk for type 1 diabetes would put it in the same ballpark as many rare diseases. We have to screen over 1,000 people to find one person that has the markers suggesting that their risk for progressing to clinical disease is high enough to enroll in a trial. So the number of people with type 1 diabetes is not all that great, but finding people prior to clinical disease is high. So just like we heard about other studies with Paget's disease, screening people who don't have clinical disease to find those who are at risk for clinical disease is a whole other world. And if you want to do prevention studies, we're in the same ballpark here. So here's an example where we don't have a treatment. This is a group of people whose 5-year, in this study, 3-year risk of developing clinical disease is about 35%. So let's say 1 in 3. Their 10-year risk is about 75%. So we know that, essentially over time, all these people are going to develop clinical disease. So we're trying to prevent the disease. We don't have a way to do that. So there are a number of agents. So here, there are a couple of agents I put on here to try to illustrate. One is an anti-TNF, so it's an anti-inflammatory agent. Baricitinib is a JAK inhibitor. And so these agents have certain properties that make them attractive and might interdict the pathogenesis of the disease. The point being here is, is what you can see from the sample size calculations, and you'll see some very clever work going on. So in the first hand, number 1, the first drug, we use a 1-to-3 randomization. So we're going to have 3 people enrolled on the treatment arm for every 1 person enrolled in the placebo arm. So that's this bias I was telling you about, this concern that people have. They don't want to participate in a trial. Well, they have a 50% risk of getting a placebo and then have to not have their disease treated. And ultimately, as I said, they have very high risk of developing clinical disease. And here they have a 1 in 4, they have 25% risk. Much more appealing, you would think. But the trick is that, since I'm going to reuse the individuals on the placebo arm, this solid line, then in the next drug, I need fewer of these people. So then the randomization ratio goes more in favor of patients with disease. They really want something, and so a higher percent, on my next slide I'll do the calculations for you, we'll get the active agent, because I don't need to enroll as many people on a placebo. And if I look at this cascade of studies, it'll all work in my favor. And if I'm trying to reduce the burden or the bias about being enrolled in a placebo arm, it's actually going to help me do this all along. And the other thing that's happening here, instead of the other designs, is I'm going to, once I've finished the accrual period for the first treatment, this is my anti-TNF, I can begin enrollment on the second while I'm following those people on the first until I see the outcome. I don't have to wait till I get the outcome before I know what to do next. I've got a candidate drug, so as soon as I fulfill the enrollment on the first drug, anti-TNF, I can start enrolling in the baricitinib trial. So I'm tiling these studies so it's going to move quicker. So I don't have to wait. Typically, if you think about one study at a time, we don't design the next study till we finish the first. But I maintain that once you stop enrolling people, you should move immediately on to the next drug. Now, when it comes to the third arm, the third trial, we can pick a new drug, or we might use the results from the first drug, anti-TNF. And so then we change the trial. But this platform design, has a very appealing property. And, so we can look at this in terms of the total number of people enrolled and the percent of people, that are enrolled on the placebo arm versus the active intervention. So you can see that, at the end of the day, I'm not using one-to-one randomizations that you've heard about discussed in other studies at all. In fact, I'm using a weighted randomization, an unbalanced randomization, to reduce the percent of people that will get assigned to a placebo. And in fact, because I have a group of people assigned to placebo already, I'm taking advantage of that for each successive study. So the unbalancing becomes even more so for new patients enrolled. The aggregate population that I get to compare remains in balance, or remains within the boundaries that I set. But for new patients enrolled, the percent or the probability of being assigned to a placebo goes down each time. So that's pretty desirable. And so we have this key feature here. Based on what we are doing, we can actually dynamically change the allocation ratio, that's dynamic allocation, so that we can test the question within the time period allotted. We can dynamically change that. It doesn't affect the sample size at all, total sample size. We can reduce the number of people, new participants that we have to enroll, overall, the total number of people, and the percent of those people that are assigned to the placebo. So the number of those people receiving the potentially active agent increases. So when we talk to patients about it, it has some very appealing characteristics. Because we're planning ahead, we're trying to look at, the future, and thinking that we're going to want to do another study. We're really not going to find the silver bullet and have a cure. We actually will want to do something next, and we're going to plan for that. And there is a caveat here, is that, more patients and active agent increases cost, right, as compared to placebo, because drugs cost money. So there's a trade-off here, patients for drug. If you can get the pharmaceutical industry to donate the drug, it's much less of a problem. If you have to buy the drug, it's certainly a problem, and we deal with that kind of an issue. For the most part in my trials, we get the drug donated by pharmaceutical industry, and they're very willing to do that. There are some, we could have a whole other presentation on, how to do research in the context of getting drugs from pharmaceutical agents, and perhaps another time going forward. Now, I do want to point out one thing, and then I'll actually come close to the end here. Let's see if I can go back here. So, the placebo arm patients really represent an interesting group, because I can enroll them de novo. I can enroll them as I go along. But if I had access to a historical database, which could be a registry, I can use the data from the registry. So there's new guidance, and I thought I had a slide on that. Maybe it will come up in a moment, where we can actually look at using historical controls. And so there's this reference I gave you from the US Food and Drug Administration that really talks to that point. So that here, rather than look at having fully enrolled prospectively patients who are receiving placebo or standard of care, whatever our design is, our control arm. If we have access to a population that is similar in characteristic, and we've got accurate, complete data, as you might have from a registry, we can use them. So we should use historical controls. And there's guidance now on how to do that. And sometimes we weight that so they contribute a little bit less than concurrent controls. There's an issue here that we have to think about in the particular disease we're studying. But what it points to me is that registry data not only helps us estimate the parameters of a trial, we know what the natural history of the disease is, but also is a source of participant information so that we don't have to now go out and enroll people for the standard of care. People are receiving the standard of care. Now we have to be careful how we do this and how we select them. And there are two ways we do this. One, we just match on the population. So essentially what we're doing is, remember, randomization is just assigning people equally in large numbers. The law of large numbers assumes that, they will be distributed. The populations on each arm will be equivalent. In small studies, we can't really guarantee that. But in fact, what we can do is guarantee it by selecting people with the same patient characteristics, maybe age, maybe BMI, maybe gender, maybe, other characteristics that we look for. So they match, our treated group. So we randomly select them from a registry historically, and we use them in their data, essentially. We don't have to re-enroll them. We don't have to consent them. We can use the data and use it as part of the analysis of the study. Now, the other approach that you heard Leanne talk about is called dynamic borrowing. And here, we can sort of borrow patients who have the same outcomes historically. So as we observe outcomes in our population, we can match and look for outcomes. Now, I'm not so comfortable with that. I much prefer to match on characteristics. But essentially, when you think about dynamic allocation, what that's doing is it's over-enrolling or increasing enrollment in the favorable group, right? So this is just the opposite that your financial advisor or stockbroker would tell you, right? So if you buy a stock and it's going up, your stockbroker is going to tell you to sell that stock because it's overweighted in your portfolio. And they'll want you to buy other stock. So as a statistician, I look at that and say, wait a minute, I don't understand that. If what I bought is really working and is really going up, why don't I continue doing that? But then she'll say, you have too big an investment in that one company, too much risk. You need to distribute. So you ought to buy other stocks that don't do as well. I have a little trouble understanding that, so I'm not good as a financial advisor. But I am good at designing trials, and we match on terms of characteristics. I don't really do adaptive randomizations looking at outcomes. We could talk more about that and discuss that going forward. But it's something to consider in terms of tying registry data, registry information to trial data. And that's a big savings if you can do that, because you can enroll fewer people. There's much less hassle with getting consenting, et cetera. Much less of a problem with respect to offering people a placebo or a treatment and participating in a trial. And they don't have to participate. Those people are being treated and being followed clinically. You don't have to create any new infrastructure. It's a lot cheaper to use those people. So try to use all the information you have. And in this sense, I recommend looking carefully at registries. And when you design registries, think about how you use the data and how you could use the data that enhances other things you're doing. I think there's an opportunity for registries that are important. to consider. And you don't just collect data for data's sake. You collect data because you have questions you want to ask. Now, in a registry context, because you have natural history data, you can do a lot of things, and we can talk about that in the future, which makes them quite valuable. And we should think about why are they valuable, because what you've done is you've got this population of individuals with rare diseases, so you've amassed them all in one place, and now you have a great picture of the natural history of the disease. People will pay for that. And your fellow providers would be very interested because, if we're looking for treatments, so where do they go for information? Well, they can go to the literature. That's good. But we actually have real data. We should actually use real world experience and try to look at that and look at people's course of the various treatments that people have tried. So one of my research programs, it deals with rare forms of diabetes. So there's the typical form, there's type 1, type 2, type 3. We're all familiar with that. There's monogenic, there are gestational. So maybe people aren't really aware. There's probably a dozen different forms of diabetes, many of which are pretty rare, right? There's Flatbush syndrome. There's, DKA-prone diabetes. There are individuals who have impaired insulin secretion, but they have no antibodies. They don't have autoimmune form of diabetes. We don't know why. Is that a cell signaling defect? There are just lots of opportunities to look at that. And we are amassing a large database. It's an observational database. It's a convenience database of patients who have those characteristics. And we can look at how well do they do under a whole bunch of different therapies and use that as a guide for the therapies we want to use. So we actually have a lot of value in data. So when we talk about rare diseases and registries, I always think about, okay, what do we want to do with the data? Let's make sure the data we collect helps us serve that purpose going forward. So there are approaches here, and in clinical trials, we can use registry data. If the registry data is carefully collected and has the same parameters or information in it than you would get out of a trial. So sometimes we do studies in trials that aren't clinically indicated, and we have to think about that. We have to think about what the consequence of that is. So, we might, repeat a CT scan every 6 months in our trial, whereas we might not have a CT every 6 months from the way we clinically manage a particular disease. We'd have to think about that. Think about what the consequences are and what the opportunity is to use the data. But I make the point that, as we heard earlier today, registries are quite valuable if we plan them well, if we think about what we want to do with the data that we collect. So I thought that, as I'm getting close to the end here, I close. I do want to provide, references for everything. So the slide deck will be available. And for people who are interested in any of the concepts that I talk about, I have them there. But I wanted to just cover just a couple other quick things in the 5 minutes or so I have left. So these are curves that kind of show you, what's the impact of different parameters on sample size. And so I put this up there to give you an idea that, this is kind of what we do. And if we want to compare the mean average response in one treatment versus the other, we do it with a formula that looks like this. And if we rearrange all these terms, that's how we calculate the sample size for that. And this is nothing more than a t-test. That's what you might expect. I just rearranged it to show you the sample size. And so the sample size is the n. The randomization ratio, if you have balanced or unbalanced randomization, you have to build that in the formula. There's the variance of variability. So we measure the mean response. It has a distribution about it. It has a variance. And these z values are nothing more than a normal distribution, the cut points that correspond to your type 1 and type 2 error. And the effect size is really the difference in the means. Mu1 and mu2 are just means. So no more statistics. You don't have to worry about that. But the point being is that as the variance increases, if you have more variability in the outcome, then the sample size goes up. So that led us to think about ways to reduce sample size by reducing variance. So holding everything else constant, if you can reduce the variance, you can reduce the sample size of a trial. So we did that. And so this is a model of insulin secretory capability. And what we did is we showed that we can model that very nicely by just taking into account the age at entry into the study and their baseline insulin secretion at the time. In fact, we could project very nicely, and there's a reference in a moment, what their insulin secretory capability will be in a year. We know how to model that. We know the natural history of the disease. So we can actually reduce the variance because this calculation, this estimate based on this model, has smaller variance because I've already taken into account, I've already adjusted for age, I've already adjusted for baseline C-peptide. So whether I'm talking about a pulmonary function test or I'm talking about an insulin secretion, same concept. If I can reduce the variability, I can reduce the sample size. If I know variables that contribute to that outcome, let's take into account that adjustment before we calculate the sample size instead of at the end of the study, because we can get by with a smaller study. And then, what we actually define then is based on the natural history of disease that we have an outcome, we know what the outcome in a year would be based on the natural history, untreated. So this opens up the door to, a calculation we can make. We can look at the observed outcome in a patient versus the expected outcome based on our historical database. So we can do this with rare diseases, we can do it with registry, because that's the natural history of the disease. So we can model the outcomes of patients the very same way. So the exciting part about this, I'm speeding up because I'm running out of time. The exciting part about all this is now we can look at a treatment effect not just by the mean, you might expect in a normal distribution. Normal distribution. There's the mean. And we can compare the means. That's what we typically do. That's what a t-test is doing, right? But if you can see, I have a whole distribution here. So I can look, in fact, at the range of response to my treatment. And you can see that some people respond much better than others. You can see the shift in this curve. So I can actually calculate that. And I can actually then look at the whole distribution of responses in the population I enrolled. Right? Because if I think about the mean, it's very much affected by an outlier. So in my population, if I have, a subset of people who are great responders, they affect the whole mean, but maybe that's only 10% of the population I studied. 90% don't respond at all. I might want to know that. I might want to be able to interpret my data that way, to look at that. And the other thing is essentially it normalizes a bunch of comparative trials. So if I had the data, I could look at a whole bunch of trials in a given disease and compare them and see which one has the greatest effect. And so I did that. And there's a bunch of studies that I ran in the prevention of diabetes space. That's what I'm looking at. And I can see how they align. I can look at their mean response and their entire distribution of responses. So I have much more information. So I believe in using all of the information we have here. And the most exciting thing about it is I can compare treatment effects at the subject level. So I can actually look at whether an individual benefited from treatment. Because remember, in the designs we've all been talking about, all I can conclude is the average response of those who received treatment might be better than those who didn't. But now I can calculate it at the individual level. Isn't that exciting? You can actually know for your patient, did they benefit and how much they benefited from that. This is a whole other layer. So I can actually look at, individual level responses, which is really pretty incredible. In this methodology we published, I think the reference up there. So The idea here, the take-home point is that, A, we ought to think about how we want to use the data and get the most out of it. And we can reuse data in different ways to reduce the total number of people we have to enroll in a trial. And in cases of rare diseases where the number of people available is limited, that's a very effective tool. Does it work? Yes. That's why we now have drugs with FDA approval that are now accepted in clinic where we didn't have them before. So we have sirolimus and lamb. We have teplizumab in type 1 diabetes. We actually can do studies that we couldn't do before because we thought ahead about our entire research plan, and we didn't consider each trial independently by itself. I'll stop there and say thank you very much.
Good. Well, I think I'll start with a closing address just to thank everybody for their participation today. It was great to meet. Met many of you last night, and really was a real pleasure. I think this conference has gone from strength to strength. Even in a year, there were about 100 participants. Last year, there's 260 people showed up today, so it really is great to see the participation from everybody. And I think that the real benefit of the multidisciplinary nature of this rare disease network, with, individually all the specialties just would be very difficult to build the kind of networks that are required to move new therapies on for these rare diseases across various specialties, from skin to endocrine to pulmonary. So it really is great to see. I'd like to thank Cormac McCarthy and Rachel and Michelle McCormack for all the organisation that went into this huge amount of work behind the scenes. They even arranged for the beautiful weather here at O'Reilly Hall for all of our visitors to see. It's typical spring day in Ireland, yes. But anyway, we'll enjoy it while it lasts. And I'd like to thank all of our international speakers and some real household names that presented today and that have been here all day. So those of you that have had the privilege of listening to them will realise these people really are giants of rare diseases, and it's great to have their participation and contribution here today. And to all of our junior investigators and the posters, it really was a pleasure to have a look at those, some really top quality work. So we have a few awards to give out. We have a couple of travel awards and then the best poster and the PPI poster. So the travel, the first travel, I'll just ask you to come up and we'll present to you. So first travel award is to IPEC Koroglu from Turkey, and it's sarcopenia, skeletal muscle impairment and Fabry's disease. So I don't know if IPEC is still here. You're not? Yes? Very good. And the second travel award is for Sarah Lopez, University of Coimbra, Portugal, which I did visit about a year ago. This really is a fantastic campus also, so congratulations. And next we're going to give the award for the best PPI poster. So that's Enhancing Huntington's Disease Research Through Public Patient Involvement, Collaborative Approach to Enhancing Research Priorities and Improving Research Outcomes. So that's Ava Woods, Trinity College, Dublin. And then last but not least is Best Poster Award, and that's to Atik Batul from UCD here. So defining the role of ATP citrate lysate in pulmonary fibrosis immunometabolism. So congratulations. So thank you all, and thank you all for persisting and staying to the end of the day. It's always difficult to stay to the very end of any conference, but I think Bruce kept people interested, so thank you, Bruce. And there's a little reception next door, some wine, where you can ask Bruce all the questions you want. So thank you all.